Greetings, everyone. On behalf of the GeneBank platform and the Crop Trust, welcome. Glad you could join us today. I'm Yanni Van Beem, and with me is Nelissa Hamora, who is co-moderator for this presentation. I'm very pleased to introduce our speaker, Dr. Julian Ramirez-Villegas. He is a principal scientist and leader of the Data Science for Climate Action at the Alliance of Biodiversity International and the International Center for Tropical Agriculture. Julian is also a professor of agricultural climate impacts and adaptation at Wageningen University and Research. He is an agricultural engineer by training with a doctorate in environmental sciences and research interests that span a range of disciplines, including climate change, adaptation, crop modeling, agrometeorology, biodiversity conservation, and geographic information systems. Julian has published more than 60 peer-reviewed articles, several in very high impact journals. The title of his presentation today is How Much Genetic Diversity is Still Missing from Gene Banks, a review of 15 years of work on gap analysis. Welcome, Julian.
Thanks, Yanni, and thanks everyone for attending the webinar. So I won't reintroduce myself. I'll be trying to walk you through 15 years, actually it's 14, of work on gap analysis that we've done collaboratively with quite a large team, quite a large international team from covering a number of organizations, including the Crop Trust and many CGIAR centers. So 2020 is the earliest target year for the SDGs. The only SDG for which a target year is 2020 is the one on conservation of biological diversity, which is SDG 2.5. And that SDG reads, we should by 2020 maintain genetic diversity of seeds, cultivated plants, farmed and domesticated animals, and the wild species. Now, it also tells us how, through soundly managed and diversified seed and plant banks at national, regional, and international level. That same SDG also talks about ensuring access and a bunch of other things which are important, but they won't be the focus of my talk, so I won't read that last part, though it is nevertheless quite important. Now, if we think about conservation, 1,750 is how many gene banks there are, at least according to the latest state-of-the-art of genetic resources conservation for food and agriculture. And 7.2 million is the number of accessions that those gene banks currently maintain. Of those 7.2 million, 2.8 are thought to be unique. Now, that's a lot, right? But let's see how that looks like over a map. Of course, a lot of what I'm going to present is going to be about geography. So maps will be popping out here and there in my presentation. First of which is going to be this one. This is how that global system, ex-situ conservation system looks like, or at least what the visible part looks like. There's of course a part which is not visible, at least not through Genesis, and so we cannot really pull it with everything else, but we know it's there. There's about 4.1 million accessions in that system. Now the question which concerns us today is, are there any gaps in what we see in there? And if there are any gaps, how big are they and where are they? Are most probably the next obvious questions that we're going to ask, right? Before I get into how we looked into this problem and some of the results that we've walk you through some of the results that we've gotten in the last 14 years of work on this very topic, I want to define what is a gap. So a gap is just, in the most simple terms, something that is missing, something that, and typically this something takes us from a current and desired state to a future desired state. I've included a cartoon at the bottom just for illustrative purposes. If we talk about genetic resources conservation, what could this missing stuff be? So it could be taxa, or it could be one taxon, right? It could also be populations or a part of the environmental range of a given taxa. of a given taxon, sorry. It could be specific samples or accessions. It could be individuals within a sample or within a population. It could be specific traits or trait combinations, and it could also be specific alleles or allele combinations that may be missing. So here I've tried to go from simple to complex, right? If we want, and of course, we can quantify gaps in any one of these dimensions or in any one of these stuff or things that could be missing. And the more we go towards the bottom of the list, the more complex and probably also the less available data we're gonna have. Before we dive into how we quantify this problem, we need to understand a bit what kind of data we have access to quantify this problem. Normally for genetic resources, we typically have three types of data. One is the passport data, which tells us where materials are from and what typically was their name, who collected them, when it was collected, at what location specifically they were collected. We also have often, this varies a lot from collection to collection, evaluation and characterization data, with characterization being a lot more common than evaluation, but both being not really very common, certainly not covering the whole 4.1 million we just looked back a few slides ago. And then we have genetic and genomic information, genetic or genomic information, and that is increasing dramatically for all collections, but it doesn't, well, there's a wide variability in terms of how this information is captured, and but it is certainly not available for all the collections. So if we wanted to look at the global system, most probably we are sticking with passport data, since we could hardly go to any levels that require anything else than passport data, right? And this is just because passport data, even if it's not perfect, is more available. It's much more available than evaluation, characterization, and genomic data, right? So what can we do with these different types of data? With passport data, we can look at taxonomic gaps, right? So if we have lists of taxa which are in gene banks and lists of taxa which are out in nature, we can use passport data to come up with those lists, and we can use, of course, taxonomic inventories in countries to compare, to use as a basis for comparison, and then understand if there may be taxa which are missing. We can do a similar type of analysis for populations, and we could also maybe a little bit, we'll go into these in the next few slides. We could also employ modeling methods to look at the environmental piece of this. Perhaps going a bit deeper, we can also look at specific accessions at specific sites. And that's what passport data allows us to do. If we have evaluation characterization data, we can also look at trait specific gaps. And if we have genomic information, we can look at the ideal level, which is the genetic level, right? Now, what is important to realize is that the main way through which we can fill gaps, if there are even any gaps, is through collecting. And collecting happens at specific sites. And you need to know where to go if you want to fill a gap. If you know that there's a gap, you need to know where to go in order to fill that gap, right? So we always, whatever method we use, we always need to go back to the geography, to a geographical component, and identify those locations where the gaps exist, again, if there are any gaps. Now, fortunately for us, everything, or nearly everything, everything in the natural world has a detectable geographic pattern. Species, populations, traits, nearly everything has a detectable geographic pattern. So we can model it, and we can come back to geography, right? But there are many buts, and believe me, after 20 years of work, after 15 years of work on gap analysis and more than 20 papers that we've published on this topic, we've heard all the buts from reviewers, from co-authors, from people who are presenting the work too. Passport data is incomplete. Not everything has coordinates. Taxonomy accuracy and precision aren't ideal. Coordinates can be inaccurate, lack precision, or just be plain wrong. There are duplicates, and those duplicates are difficult to detect. locality information isn't very complete either if we want to georeference accessions which have missing coordinates. And there may be more, right? But that shouldn't prevent us from trying to get an understanding of the problem, whether we are conserving enough diversity or whether we are, or how close we are to the target of full conservation of plant genetic resources. So now let's take a look at the problem, right? This is how we did it. And we devised two different methodologies, one for wild species, another one for domesticated species. We specifically in the domesticated gene pools, we looked at land races only, of course. Because those are the ones which are at most risk of genetic erosion, right? And so I'm gonna try to walk you through the two different methods, right? So now let's take a look at the wild methods. The first question we asked for the wild species was we wanted to know which taxa are sufficiently conserved or well conserved. Sorry for the typo up there. This method we published in 2010, but actually the work we started around 2008. So that's where the 14 years come from. We try to answer this question about which taxa are well conserved through three simple questions, or seemingly simple questions. Is the number of samples in gene banks comparable to what is known about a particular taxon of interest, right? We came up with a metric called the sampling representativeness score, which compares the number of samples in gene banks with the total taxon observations, which we can get from many sources, including but mainly say from herbaria. The second question we ask is the geographic distribution of these samples representative of the distribution of the taxa, of course, needless to say. And if you look at this blue square here, if you imagine that blue square is the geographic distribution of the taxon and that the white spots are the samples, the germplasm samples, we can try to compare the area which is represented by those samples with the total area, with the total blue plus white area. That's what we call the geographic representativeness score. And then lastly, because we're also interested in the environments that those samples cover, we ask whether the environmental distribution of these samples is representative. So this we do by classifying the environments where the taxon grows. And in this graphic, an environment is ascribed by each of these squares of different colors. So orange, yellow, gray, and green. And whichever of those squares, climate type, namely climate types, ecosystems, or environment types, doesn't have accessions represented here by the red dots, then that would be considered a gap. From these, we calculate something called the environmental representativeness score, which tells us how many different environments have accessions versus how many environments the taxon grows in. And then from these, we come up with an aggregate score, and we can then classify the taxon as being well represented in gene banks, or less well represented, poorly represented, or extremely poorly represented. We have 4 categories actually. Then the second question we ask is for those taxa, for any taxa which aren't well represented, then we would like to know where the gaps are, of course. And this is where the modeling of species distributions comes in. I'm not going to dive into those details. One important thing here is that we're using geography and environment as a proxy for genetic variation, which may be completely wrong, but it's the best we can do at this point. And I'll come back to this issue at the end of the talk. Now let's look at the domesticated before we go into the results. The first thing that we look at, the domesticated gene pool is often quite complex, but we know more about the domesticated gene pool. There's generally much more accessions comparatively for a given taxon compared to what's being collected for the wild relatives. And there's a lot more studies, especially genetic studies, that help us classify understand a little bit more about the genetic diversity within that. So instead of going at the taxon or at the crop level and conducting gap analysis at that level using the wild relative method, we wanted to develop a method that recognized what we know about the genetic variation of the domesticated species. And so we tried to, what this triangle represents is a schematic basically of that genetic variation. At the very bottom, you have the most disaggregated level of genetic variation that can be ascribed to a land race, which is the individual land race. Unfortunately, we almost never have that kind of information from any gene bank database, and certainly not across the board. So we are somewhere in between. What we do is we choose a level in between that we know responds to socioeconomics and the environment, and then we stick with that level and look at gaps. So now, how do we look? So, for example, for sorghum, we look at sorghum bicolor races. For beans, we look at bean, Andean, and Mesoamerican gene pools, and we do the analysis at that level. Now, what analysis do we do once we can identify this level? The analysis that we do helps us understand which locations in the geographic distribution of a given landrace group, call it Mesoamerican beans, are a gap. To do this, to identify which locations, we ask 3 questions. Is the site that we are looking at easily accessible from any other existing collection site? The second question we ask is the site well within the network of existing accessions? And then the third question we ask is the site environmentally similar to any other accession in a given climate type? So we ask about about the geography, the nearness, right? The geographic closeness. We ask a question about the density of the network of accessions, and then we ask a question about the environmental representation. And with this, any areas which do not meet any of these criteria, they will be considered high confidence or high value gaps, right? This is what you see in the map here in red is the high value gaps for Andean beans. But now I'm getting ahead of myself. I'm giving you part of the answer, whether there may be gaps or not. I apologize about that. Before getting into that answer, I want to tell you where this answer comes from, where this answer, at least the answer that we think that we got so far comes from. There's 2 multi-crop global studies that we've conducted, plus a number of other papers that the team has published. The first one, the first one of these global multi-crop studies is Castañeda Alvarez et al. 2016 published in Nature Plants, which analyzed about 1,000 wild relative taxa related to 81 domesticated crops. And there's a second study, which is under revision right now, also in Nature Plants, where we looked at 72 different landrace groups of 25 domesticated crops. These are the 2 main studies that I'll be talking about. Nelisa and Yanni are sharing 3 methodological papers that we've based these 2 studies on. And as I said, if you look at the Google Scholar of Colin Curry, Luigi Guarino, myself, Nora Castañeda, you'll find the rest of the papers one way or another. So now let's get to some of the results. So for crop relatives, what we found is that about 71.1%, exactly 765 species taxa are high priority for collecting. And on the other end, only 4.2 or 45 taxa are sufficiently represented in gene banks. Now let's look at this in a little bit. Let's flesh this out a little bit more. Here you have the crop groups in the y-axis and the number of crop relative taxa that we analyzed at that time. And you can see that there's differences as to the number of crop relative taxa for each of the different crop types. But regardless of the crop type, there's a lot of red, and red means high priority for collecting. There's very little green and purple, which is low priority for collecting or collecting not required. So have we done enough for crop relatives? No, not really. we haven't done anywhere near enough across the board. As I said, the broad global number would be that 70%, around 70% of crop relatives of 81 of the most important crops for food and agriculture are not really well preserved. Now let's look at land races. What you see here in this scatter plot is on the y-axis, the degree of crop relative representation. I chose to keep that just so everybody gets a sense of the relationship between one and the other. So that's what we saw in the previous slides, but now you can see it at the crop level, not at the crop group level, right? So degree of crop relative representation. And on the x-axis, you see the degree of landrace representation in ex situ conservation. Now, the first thing that becomes obvious is that for land races, well, it depends, right? So whereas for wild relatives, you really have poor conservation across the board. Some more than others, but conservation is really not ideal in most cases. In all cases, for land races, it depends, right? You have crops like grass pea, wheat, chickpea, which are relatively well conserved. And you also have crops in the other end, like pearl millet, yam, and finger millet. Now, on average, gene banks conserve the equivalent to 63%, this is according to our estimates, of the landrace geographic distributions that we analyzed of these 25 crops. At the crop level, though, one interesting finding for us is that landrace conservation levels are not quite correlated with crop relative conservation. So crop relative conservation is quite poor compared to landrace is generally higher. Now, if we want to flesh that out a little bit more and look at the scale of the problem, this slide lets us do that a little bit in a little bit more detail. What this slide shows is the crops in the y-axis and then the amount of area which is considered to be a gap in million square kilometers. This is just the physical area that's in the x-axis, and you can see all crops sorted by the area of crop relatives. The first thing that is very obvious is that the area of crop relative gaps is much larger than the land races area, but you can see that even even for the land races, in some cases we're talking about a few million kilometers square that may need collecting, right? This gives you an idea of the scale of the problem. I'll come back to the scale in the conclusions, right? The second question is, where are the gaps? So we looked at the regions. One of the things we did is we looked at the regions. What we're doing right now is we're looking at the regions, the difference between regions. What we see is that in both crop relatives and land races, in both cases, we see Africa, Asia, and the Americas as having the greatest amount of physical area, which is considered unconserved, or which is considered a gap. And conversely, we see Europe and Oceania as having much less area, right? But we can flesh this out a little bit further and look at a global map of gaps. Before we do that, it's important to understand what the diversity pattern is. So this is just the total richness as the number of crop gene pools, which are predicted to occur in any particular location, and compared to the number of landrace groups, which are predicted to occur in any given location. Those two dimensions are illustrated by two different colors. The oranges are used for the crop relatives, and the blue-green is used for the land races. Now, when you see a dark blue-purple, which is around here, you, like in Spain or like in Turkey or like in the highlands in Mexico, you see that both of them are occurring at high levels of diversity. Now, what we're finding is, in spite of some documentation issues, because we don't have, we cannot see national gene banks of some countries, and for example, China is one of those cases. In spite of some documentation issues, our models actually capture the centers of diversity and domestication pretty well. Now let's look at the gaps. The other thing is there seems to be some correlation in total diversity, at least for some countries or some regions, right? Now let's look at the gaps. So let me go back to diversity map and go back to the gaps. So consistent with, of course, what we saw before, we see a lot of purple, which means that there's a lot of gaps for crop relatives. There's a lot of species of crop relative taxa which have gaps. Conversely, where we don't see purple, we see a lot of gray, which means that both land races and crop relatives there's less taxa basically, or there's less land race groups which have gaps. In some specific spots, they're hardly noticeable here at the scale of this map, but they will be if we zoom in. I'm not going to do that here, like in Turkey or like in Mexico or like in some spots in the Andes. We will see this dark purple, which is areas that have many taxa and many land race groups with predicted gaps. So now we understand at the global level what is the extent of what is the extent of conservation, how well the global system is doing, at least in terms of how much of the diversity that's out there, assuming the diversity is correlated with ecogeography, of course. Now we can start asking a lot more questions, right? So one of the questions is, what are the environmental patterns that are most often missing? What diversity patterns emerge and at what scales are these consistent with domestication and diversity centers? Which diversity or domestication centers have most gaps? What about gaps? We can also ask practical questions about specific crop gene pools. What about gaps in specific crop gene pools or in specific races within given crop gene pool, say domesticated gene pool? Can we see gaps for Mexican highland maize? What countries are most priority based on uncollected area is also a very interesting practical question if we wanted to start thinking about collecting, and there may be many more. If anybody here in this room wants to work in any of these questions, feel free and wants to use the data that we have, feel free to reach out and we'll happily share what we have with you. So one of the things that we found is, of course, we haven't done enough, but should we be collecting more? Now, there's several perspectives we can take on this question. The first one is a purely conservation perspective, which reads more or less like this. Diversity is a risk, it is unpreserved, and we need to preserve it. Thus, we need to collect urgently. Now, there's a second perspective, which is a use perspective or a measured use perspective, if you want, which would read more or less like this. Large parts of collections are still unused by researchers and the general public. Before investing in more collecting, we may need to first understand if we have what we need, for example, for breeding climate adapted crops. Now, you can go back to, if you want to have an argument, you can go back to the conservation perspective and say, well, if we wait too long, until we know if we have what we need, maybe when we realize that we don't have it, we go out there and collect and we find nothing. So anyway, these are some of the questions that we should be asking ourselves. Probably the answer is quite crop and gene pool specific, maybe taxon specific. In any case, if we go back to my very first slide on the SDG target, and we look at these numbers, we're nowhere near those targets. So we should in many ways at least be asking whether we should be collecting and where. Now, if we want to collect, a key question is, does collecting really work? Does it fill gaps? We know that there's many gaps, 70% species, blah, blah, blah. Would collecting actually help us fill gaps? Now, we know that gap analysis can support collecting. Here you see an example of gap analysis outputs for sorghum and African maize on the left, being pulled together to draw a collecting route in South Sudan. And this information can be provided to collecting teams who can then discuss amongst themselves and plan collecting and go to the field and actually collect the material, right? But if we do it in this way, will we be filling gaps, right? Now we have a recent experience from the crop relative project, which the Crop Trust has been leading for the last 10 years. that demonstrates that if we do targeted collecting in areas of gaps, the collecting actually does fill the gaps. So the crop relative project, and thanks Nora to Nora Castañeda for sharing these preliminary results with me. The crop relative project collected around 3,000 new crop relative samples in around 22 countries. You can see the geographic distribution of those samples as green dots in that map. Now, Nora and her team, they're asking this question, the Crop Trust team are asking the question, are we filling gaps? And the answer is actually, for the most part, yes. These 3 examples, one for apple, bambara groundnut, and the last one for sorghum. What you see in the y-axis is the representativeness in gene bank score. This is, just take it from 0 to 10. This is the degree, how well a taxon is preserved. If you can see the gray is the individual taxa that have been analyzed, which is the taxa that have been collected. And the red is the average between those. You see 2 time periods in the x-axis for every one of the, for each one of the crops, 2014, 2018. And what you see in each case is that the red line has a negative slope, which means that the conservation, the representativeness score is going down. Now, a high representativeness score here means that the taxon, or in this case, in the red line, the average of those taxa, it's not, those taxa are not well represented in gene banks. When you have levels which are at or below 2.5, that means that the degree of representation is quite well. And both in average and as well as individual species, after the collecting, the degree of representation increased quite substantially. Only in, if you can see it here, in the case of one of these taxon in apple, the slope was not so strong. But in general, we can say that for the majority of taxa that have been, for which targeted gap analysis-based collecting has been done, there is an increase in the degree of conservation. So we can fill gaps. It may not necessarily be very cheap, but we can. So now some conclusions. After 15 years of hard work, we have methods to predict conservation gaps in wild and domesticated plant genetic resources. Now, this comes with a few buts in terms of data, but also in terms of method. One of our biggest assumptions here is that genetic diversity is at least in some way correlated with ecogeography, right? And this may not hold true in all cases, but it's the best we can do with the current, at this level, in such comprehensive way, and with the data that we have. Now, despite the efforts to salvage plant genetic resources, especially during the 1970s and 1980s, one of our main findings is that gaps exist. 2/3 of crop relative taxa are in need of conservation. And for many of them, we know very little, but we know that they may be endangered. Land races are much better represented, with 63% of their geographic area currently represented in gene banks. And collecting, if done smartly, it can help filling those gaps. But, and again, I want to come back to the scale of the problem. If you remember those bar plots with the area, the scale of the collecting needed appears to be quite large. Lastly, I wanted to bring out a point about our information systems and knowledge. After all these years of work of trying to publish papers on this topic, of trying to model landrace distributions, crop relative distributions, and looking at the whole, at the global picture in terms of data, we also have some conclusions around this. And one of them is that data updates and data quality definitely need improvement. We've spent a lot of time trying to make sure that what we produce in terms of modeling makes sense. And that means spending a lot of time curating data. Now, if this data would have been curated to start with, it would have been a lot easier for us. Now, this is not a complaint necessarily, but what we think is that what we have observed from all of our modeling results is that better data improves the accuracy of any estimates of where to collect. So if we have estimates of gaps, if we want to use those estimates of where those gaps are to target collecting good quality, reliable passport and other data, will help us reduce the risk that we're going into the wrong places, essentially. Now, data management during and after the collecting can lead to us not knowing whether gaps are filled or not. I know that this is a challenge that happened during the gap filling effort of the correlative project. Now, I don't want to get into those details, but this very issue creates an risk of inefficiency that we go collecting twice into the same place. New methods that mine genomic data. One of our biggest assumptions is that ecogeography is correlated in some way with diversity, which, as I said, it may not necessarily hold true. So we need to come up with new methods that mine genomic data and help us get past ecogeography and any assumptions related to it, especially about the diversity that we have and that we don't, and its potential use, right? So there's a whole new world out there to explore if we start looking at, if we go beyond the ecogeographical data into the genomic world. Finally, we need ways of monitoring risk of genetic erosion, right? So what I provided here is a global picture. And in many of these areas where our analysis and models are predicting that there are gaps, some of them may be exposed to certain risks of genetic erosion. Some of them may be less exposed to those risks. And understanding what risks there are and how they vary geographically, which gap areas are exposed to more risk than others, this will help prioritizing even further and would help perhaps keep this balance between the sort of pure conservation perspective and the use perspective on collecting. And I wanted to end with this picture, which reflects the extent of urbanization around some area in the Nile. You can see from 1984 to 2021 how much the agricultural landscapes have changed, not necessarily to stay as agricultural landscapes with different crops or with improved varieties or whatever, which also happens, but to change to transform essentially to an entirely different landscape. So whatever has been uncollected, left uncollected or is not represented from 1984, a lot of that may have already been transformed into urban areas, and that diversity may not be salvageable anymore. So I wanted to end there, and thank you, everybody. I'm happy to respond to any questions should you have them. Thanks.
Thank you very much, Julian. That was a really nice walk through all the things that we still have to do, especially collecting in crop relatives. We have several questions, and if it's okay with you, I'll just read them out and you can answer as they come. The first one from Vinod Kumar. Most of the CG gene banks and national gene banks have poorly represented wild species in their collections. How do you justify or get around doing a gap analysis with incomplete information on occurrence and distribution of wild species?
All right, thanks, Vinod. This is a very important question. This is part of the buts. There's part of that question which I cannot possibly answer, right? So there is this. What that means is that there's uncertainty in our estimates, which comes from the fact that not everything has a geographic tag in it. Nevertheless, the methods that we have developed try to account for that to the extent that we can. But again, we go back, we rely on what information is available to us, and if the information is not good, it's not enough, then we can come up with ways of dealing with that. But there's a point where we just can't do anything, right? So, for example, to give you a couple of examples for the relative method, we introduced this sample representativeness score that just looks at how many samples are there in gene bank, how many accessions are there compared to how many observations in total, without looking necessarily at if anything has coordinates or not. precisely to try to reduce the noise from the fact that, or try to account for the fact that not everything has a geographic tag, for example. Now, there's also taxonomic issues, right? Taxonomy has changed over time, and then not everything in collections is fully taxonomically accurate. We try to work with curators as much as we can. We try to revise the taxonomy. We review the taxonomies. We look at the synonyms and we do our best, but there's a level at which we cannot go because we just don't know, right? So this is why my plea is, if we want better estimates, we also need to have better data.
Okay. From Ali, we have the following question. How to be sure, it's along the same lines, that the gap analysis is not statistically biased based on the data and the information that are used to predict such gaps?
Yeah, thanks, Ali. This is also a very good question related to the brief, so I won't repeat what I said. So in our method, I didn't go into this level of detail, but in our method, we build in a specific step to where we extract information from the collection, we remove information from the collection, and then try to see if we can predict that as a gap. In other words, we create an artificial gap in the collection and then see if our method can predict it. This way we can make sure if the method actually works or not. The other way that we know it's working is also because of the last few slides I was showing the targeted collecting actually using the information about what we know about gaps. It actually helps. It actually shows that we can increase the representation in gene banks, right? So that also gives us confidence that what we're producing is not noise. But as I said, and I'll repeat it, the models are clearly not perfect, and they would benefit from more comprehensive and better information always.
Right. We have two related questions, one from Manuela and one from Martin Van Ginkel. So the one from Manuela is like an opening statement. Thanks for your great talk, Julian. I understand that you integrate eco-geographical taxonomic passport data. Will you be able to, or have you already integrated genetic data into the model? And if yes, how?
No, we haven't. Just to answer plainly, nevertheless, we have some ideas how to do it. And we have discussed this with a few people. There's not really an easy answer to this, but it's a very interesting research question. So one of the ideas is that in one of the metrics could also be not only the geographic distance, environmental distance, but also look at the genetic distance. Now there's challenges with that, and we would need to figure out how to create a reference that will allow us to look at the genetic distance. But that's one of the ideas that we have, but we haven't done it so far.
Okay.
It should be an interesting area to explore.
Yeah, I think so too. Related to that, Martin Van Ginkel poses the following comment. Genetic diversity score should be another measure for gaps. Genetic analysis of existing samples would identify which regions contain the most genetically diverse samples. There, more collecting should be prioritized with a higher chance of finding additional genetic diversity.
Yeah, this is a very good idea, Martin. I agree, and this is some of the things that I think could be explored if we bring in genetic genomic information. Fortunately, there's a very clear trend towards increasing that kind of information for a number of collections, right? And so we could have a very good starting basis.
Okay. From Teshome, we have any explanation why sorghum landrace is one of the poorly conserved cereal crops? Looking at ICRISAT and other collections in Genesis, one may feel that it is very well conserved.
Yeah, Teshome, this is a difficult one for me to answer because I'm not a sorghum expert. From looking at because ICRISAT has also published a number of studies on gap analysis. From looking at those studies, both for relatives and also for the domesticated gene pool, there are definitely gaps. So yes, there are a lot of accessions, but but the geographic distribution and the environmental range of sorghum out there is also, and this wild sorghum taxa is also relatively wide, and that means that even if there's a lot of samples in ICRISAT, which I know there are, of course, you know that better than me, that even if there's a lot of samples, that just means that they are not enough to cover what's out there now, at least based on ecogeography, right?
Yeah. From Theo Van Hintem, thank you, Julian, for a great talk. The cost of ex situ conserving crop wild relatives is generally very high. Conserving them in situ, possibly with an ex situ backup and a monitoring system, would be preferable. Would you agree with that?
I don't know. Actually, I don't know what the costs are of each system. I know that based on our, because we have also looked at in situ conservation, I know that at least for all of the plants which human beings use around the world, in situ conservation actually is by quite far covering more diversity in terms of number of taxa and number of environments, right? Nevertheless, I wouldn't be able to say anything about the cost, right? What you suggest seems to be sensible, but and what I read almost everywhere is that they need to be complementary. Nevertheless, I mean, what you're suggesting seems to be a good way that the two efforts can be complementary, right? There's a fundamental challenge with in situ conservation, especially in developing countries, though, as to how to make sure that it actually works. But again, I'm not an expert in that particular area, so I will refrain from diving more.
Yeah, thanks, Julian. The next one is from Olaniji in Nigeria, and it's about some flagship crops or lesser known crops. Thanks, Julian. Does the gap analysis take into account some minor but important food crops that are not represented in any of the gene banks? Such species could be more endangered than the prominent crops being considered by the analysis in most regions.
Yes, so for the wild relatives, we covered quite a wide number of crops. For the land races, we did not cover, we only covered 25 crops, which is actually one of the questions we're getting asked by the reviewers. Why only 25 crops? Well, clearly they don't realize how much effort it takes to model 22 to do it with exactly. But anyway, it's a fair question, and we have some estimates. So actually, we have some estimates. I could pull them out real quick that help us look at the importance of the crop and the extent of conservation. The quick answer is that there's no real clear correlation between importance of the crop and extent of conservation for land races or for wild relatives. Nevertheless, you see crops which are highly important with a lot of gaps, and also crops which are say less important, underutilized, you can call, or minor crops, which are also not very well preserved. And you basically see the whole spectrum, right? There's not a clear correlation for both land races and crop relatives. These two global papers that I mentioned have some estimates of that.
Thank you. Does the diversity available in the gene bank represent the diversity existing in nature at taxon level? If not, what percentage does the gene bank represent diversity existing in crop species in nature? What percentage of the taxon in the gene banks is represented in nature?
So the answer is no, it doesn't represent all of the taxa which are out there. There's many taxa, there's several taxa that we flag as poorly conserved just because they, at least in the gene banks and the gene bank databases that we looked at, there is just no accession, right? So I cannot tell you an exact number now, how much that is, but it's part of the 71% for the crop relatives, is part of the 71% taxa which are in originator conservation. A big part of that is actually some part of that maybe reasonably big. You could say 20-30% of that is a taxa which don't have even one sample in gene banks currently, or at least at the time the study was conducted. For the domesticated gene pool, all of the taxa are there, yeah, with at least one sample. So that's less problematic. When you start diving a little bit deeper and deeper into the genetic structure of the domesticated gene pool, then that answer becomes a little bit more nuanced, and there may be variations depending on the country and the specific say subgene pool or group or genetic group that you're looking into.
Okay, so this one has to do about highly cross-pollinated crops. How about collecting diversity in highly cross-pollinated crops at regular intervals by using previous data of collecting sites and the pattern of diversity exists at present? So a comparison of in cross-pollinated species, this person is suggesting collecting at certain intervals to see whether they're gaining or losing the diversity that you're hoping to have in a particular region. I'm expecting that the question refers to.
Yeah, this is a good idea, Vinod. And I think there's some studies that have done this and looked at the diversity, looked at changes in diversity in time. In fact, this year, we had an opportunity. Unfortunately, we couldn't quite capitalize on it. We had an opportunity with sorghum in West Africa, where people were going out and collecting, and there had been collecting missions or at least field visits that characterize the genetic diversity of the land races in a number of places that were going to be visited. And that would allow for a comparison. Unfortunately, we couldn't do it ourselves, but there are groups that have done that. And yeah, it's an interesting thing. Whether that helps us define a frequency, an optimal frequency, that remains to be seen. I would imagine, yes, we could do that. But given the size of the gaps that we see, and the fact that it's already hard to get collecting going just at all for certain places, I think that may be quite far down the line, thinking about frequency.
The next one is really interesting. It's Chris Richards. Nature determines where crop wild relatives grow, whereas humans determine where land races grow. So I think the question here is, does your model take into account that people are planting land races and you can physically decide, which gaps to fill and send stuff out with land races, whereas you cannot with crop wild relatives? And I think this one, and then it's related to another one which is still open. So maybe we'll take this point about the human decision point. Is the gap analysis model considering the human decision point?
Yes, it is. Well, depending on what you mean by decision, right? But yes, we consider anthropogenic factors, right? So for the crop relatives, and by the way, thanks, Chris, and we've discussed this a number of times with Chris and a few others back in the times when sharing physical space was still possible. So for crop relatives, we consider only the climate and soil drivers of the biophysical drivers of their distribution. For land races, we integrated a number of additional drivers that help us, that basically bring the human element, the human choice element, or the fact that the geographic distribution of land races is shaped in some proportion by climate, but it's also in some proportion by biophysical factors, and in some proportion by humans and by their socioeconomic characteristics and preferences and whatever, right? So what we have included is things like access to market, access to irrigation, ethnic diversity, language diversity, at least to the extent that we have geospatial data to do so. Now, there's probably significant improvements that can be done if we had better data, but certainly now I'm trying to think of things like the Facebook wealth maps, right? That could be an interesting driver to put in there. But but we have considered that difference. We use similar type of modeling approach, so species distribution models, but we have a range, different range of drivers.
Wonderful. And I think I'm going to summarize these three questions that are left are kind of on the same plane. So let me summarize. Do you think that reliable data on wild species distribution is going to be available or is available now? If yes, what should be the priority to explore an in situ conservation strategy? Should there be a strategy going forward? And what would be your recommendation? What is your advice for everybody to go on? Where do we go? How do we go from here? What would be your advice?
So, okay, so in situ conservation, I don't know, my talk was not really about in situ conservation. There's an interesting paper on in situ conservation that that I published with Colin Curry. Colin Curry is the main author of that paper. I can forward that, and it provides some conclusions as to the complementarity between ex situ and in situ, and it creates a metric that assesses the extent of conservation, and it provides some general recommendations, at least as to what needs to be done. Now, for ex situ conservation, what we, together with the Crop Trust and other CGI and CGIAR colleagues, we have come up with a global level prioritization of countries. And there's document, I think I could probably share the current version of that document. Peter Giovannini, who I think is here, helped, and Luigi Guarino, who's also here, helped develop that approach. What we did was we tried to prioritize countries based on two main criteria. One being how much area for gap area is there, and the other one is roughly how many different crop gene pools would need to be collected. We included a bunch of additional criteria, including whether there are well-maintained national collections and things like that, and came up with a list of priority countries. That list is available, and that would be a starting point for any plans on further ex situ conservation. As part of the GeneBank platform, collecting missions were set up in a few of those countries. For example, Niger, for example, Chad, Togo, and a few others. And led by CG centers in the different countries, and that's an ongoing effort. There's an ongoing effort for collecting. So to answer, well, that's a long answer, but that prioritization document is probably a good starting point. I didn't include that here. Probably I should have, but anyway, the document is available, and it's probably a good starting point. We will be trying to write a paper out of that document in the coming few months.
Thank you, Julian, for walking us through a very, very fascinating study. We wish you all the best coming through. I think this is really good advice, and it gives us a place to start, at least, taking action of, regarding some of the things that you've recommended. So to the Crop Trust staff and colleagues, thank you very much for your support behind the scenes for this webinar. And to all webinar participants, we wish you a very happy holiday and a joyful new year. Thank you very much again for attending.
Yanni, I think
before everybody goes, yes. let me sorry to interrupt you, Yanni. Before everybody goes, I just wanted to, on behalf of everybody, thank you for your amazing work during this last year, organizing the GROW webinars, and to everybody, I'm sure everybody will join me in thanking you. I'll tell everybody that we hope to keep these going into the new year, and we hope to see you soon. Bye, everybody.
Wonderful.
And thank you again, Yanni.
Thank you. Thank you, everybody. Thank you, Julian.
Thanks, Yanni. Thanks, everybody.
Bye.
Thanks, Julian.